Hello, my name is Joey Peters, and my project is regulation of the BMP receptor Wnt in Drosophila melanogaster. So the Van der Zalm lab studies neuronal development in fruit flies, specifically at the neuromuscular junction. This is the region where motor neurons innervate skeletal muscle cells. The amount of growth, or lack thereof, is quantified by the number of terminal axons, or boutons, on these motor neurons. My project is studying a specific gene called Wnt that we believe acts in a signaling pathway responsible for neurodevelopment. My hypothesis is that tricornered, a gene previously identified by our lab, phosphorylates Wnt to inhibit synaptic overgrowth. In order to study this hypothesis, two mutated Wnt constructs must be made. A version that acts as if it's always phosphorylated, and another that acts as if it's never phosphorylated. And in order to generate these constructs, we needed to use an at-p scar, and this allows for site-specific recombination. This site-specific recombination allows for generation of those two non- and always phosphorylatable versions of Wnt. The at-p scar was generated using CRISPR-Cas9 technology. CRISPR-Cas9 utilizes a Cas9 enzyme, which is a bacterial enzyme that complexes with a guide RNA. And essentially, the guide RNA tells the Cas9 enzyme exactly where to cut in the genome. Then, after this double-stranded break has been enacted, homology-directed repair ensues, and a piece of DNA that we have already synthesized is inserted into the gap. This piece of DNA, or we'll call it the homology-directed repair assembly, contains the at-p scar. So in order to generate the homology-directed repair assembly, 4 distinct DNA segments were amplified via PCR: a left Wnt flank, a right Wnt flank, a plasmid backbone, and an insert containing DS-RED, ampicillin resistance, and the at-p scar. These were then confirmed by gel electrophoresis, and then using specific primers, their overlapping ends were annealed, ligated, and transformed into E. coli to be replicated via an HDR assembly reaction. In order to generate the guide RNA, a site-directed mutagenesis reaction was used. This generated a small substitution in a previously synthesized plasmid. This small substitution would be homologous to the anticipated cut site in the Wnt region. So this would allow Cas9 to cut at our specific cut site. The guide RNA and homology-directed repair assembly were then analyzed by sequencing. We can see a chromatogram picture that visually represents confirmed base pairs of each construct. By cross-referencing these to the published Drosophila genome, we essentially ensured that no spontaneous mutations occurred in these plasmids. After this confirmation, both the guide RNA and the homology-directed repair assembly were injected into a fly that was already expressing Cas9. To ensure that CRISPR-mediated homology-directed repair occurred, we used the DS-RED selector, which was found in the insert, and this is not normally expressed in fruit flies. What it does is generates red fluorescent-eyed fruit flies. This allowed us to visually select which flies underwent CRISPR-mediated homology-directed repair and which did not. So the next steps involve generating yet another plasmid, the at-b plasmid, which will be placed into the Wnt gene at the at-p scar, and this is what will finally generate those phosphorylatable and non-phosphorylatable Wnt constructs. This will allow us to count the number of boutons in these mutants to determine if significant synapse overgrowth or undergrowth is present with or without tricornered. I would like to acknowledge the Collar and Weaver Summer Under Research Fund, the John Carroll University Biology Department, the entire Breuer Lab at Case Western Reserve University, National Science Foundation, and Caitlin Tutoret. Thank you.